Hi everybody, Dr. Biology here. This is Revised with Dr. B. This is week 1, cells. How it works. So what I'm going to do is go through the main points of a topic, in this case cell biology, and not everything, just some of the important bits. You'll be given some signposts, further resources, particularly detailed videos and past paper exam questions, and there'll be weekly videos up until your exams, so please do subscribe. Firstly, animal and plant cells. So they do like you to be able to label an animal and plant cell and know the differences. So plant and animal cells are known as eukaryotes. The similarities are between them. They both have a nucleus, cytoplasm, cell membrane, mitochondria and ribosomes. The differences are that the plant cells only have a cell wall, chloroplasts and a permanent vacuole. Bacteria or prokaryotes. Their differences to eukaryotes are that they have no nucleus, no chloroplasts, no mitochondria, and they are smaller. You would need in this section to know about the required practical how to use microscopes. Describe how you would set up a microscope to examine a prepared slide under high power for 6 marks. So the answer should include the following points. 1. Select a lower power objective lens. 2. Move the stage as close to the objective lens as possible. 3. Place the prepared slide on the stage. 4. Focus away from the slide using the coarse focusing adjustment until the specimen is in focus. 5. Move the high power objective in line with the slide. 6. Use the fine focusing adjustment to bring the specimen back into focus. And 7. If the specimen cannot be brought back into focus, refocus under low power and repeat stages 5 to 6. They'll also expect you to know about specialized cells, so nerve cells, sperm cells, muscle cells, root hair cell, xylem and phloem. Key things are structures, so nerve cells, they're long branch connections, they're insulated and they transmit electrical impulses. Sperm cells got a tail for swimming, they've got lots of mitochondria, they're streamlined and they fertilize the egg by swimming to it. Muscle cells, they're long and elastic, they've again lots of mitochondria and they contract and relax to allow movement. Root hair cells, they've got long extensions, so large surface area, they have no chloroplasts and they absorb water and minerals from the soil. Xylem cell is hollow or strengthened with lignin, transports water and minerals up the plant, and the phloem cell contains sieve plates, they are living cells and they transport sugar and nutrients around the plant. You'll also need to know about light versus electron microscopes. So the light microscope has a low magnification. You can view live specimens. Samples are prepared easily. It's less expensive and portable, but you have a lower resolution. Whereas an electron microscope is higher magnification, does require samples to be in a vacuum, therefore they're dead, and they're often coated with metal. It's expensive. It requires special lab environment. It does have a higher resolution, and as it says there, cannot view live specimens. Similarities are that you can view small objects not able to see with the naked eye. You'll also need to know this, the AIM triangle or IAM triangle, to work out actual image size or magnification. You'll need a ruler and you'll measure a picture in millimeters. Now, what they do like you to do is to convert millimeters into micrometers and sometimes into nanometers. All you have to remember is to times it by 1,000 each time. So millimeters to micrometers times 1,000, micrometers to nanometers times 1,000. You also might be expected to show standard form. Here's an example. Suppose the image of a cell is 8 millimeters in length under a microscope with a magnification of times 1,000. So to find the actual size, it's 8 millimeters divided by 1,000, gives you 0.008 millimeters. Times it by 1,000 to get 8 micrometers. Cell division. Cells divide, so in the cell cycle, when an organism grows or it becomes damaged and needs to produce new cells, what does it do? It produces genetically identical cells. So if we talk about mitosis, It's really important. Don't use any names of the phases of mitosis. You sometimes see that in revision videos. Do not do this for AQA. So these are the stages. I want you to learn these stages. So DNA replicates or duplicates. One set of chromosomes is pulled to each end of the cell. The nucleus divides, so two nuclei form. Cytoplasm or cell membrane divides, which is known as cytokinesis, and two genetically identical cells form. Stem cells, what are they? They are undifferentiated cells that can divide and differentiate into specialized cell types. They're essential for growth, repair and development in organisms. You find them in two places. Embryonic stem cells, they can differentiate into any type of cell, whereas adult stem cells, they're found in certain tissues. In this example here, it's bone marrow. They can only differentiate into a limited range of cells. As you can see from bone marrow, you get stem cells that produce red blood cells, platelets and white blood cells. You might be expected to know the arguments or evaluate the use of stem cells. There's a really good list. I'm not going to read out this complete list, but you will need to know the pros and cons. So that's an evaluate question. So you could pause here if you want to have a look at those. Transport across cell membranes, you need to know about diffusion, osmosis and active transport. Here's a very good, useful summary table about each and some examples. Again, what I would do is probably pause and actually note this out, please. They do like you again to know key differences and similarities. So diffusion and osmosis are passive, in other words, no energy is required, while active transport does require energy. Diffusion and osmosis, they move substances down a concentration gradient, but active transport moves substances against the gradient. Osmosis only applies to water, whereas diffusion and active transport involve various substances. Key similarities all involve movement of molecules. All occur across cell membranes. They're all essential for cell survival and homeostasis. There is another required practical on osmosis, looking at the effects of salt and sugar concentrations. Again, you can watch my video or another video on that. So osmosis occurs from a dilute solution to a more concentrated solution across a partially permeable membrane. The higher the concentration of the solution, the more water is lost from the potato cells by osmosis. You'd be expected to know about surface area to volume ratio, and it's a measure of how much surface area an organism has relative to its volume. So this affects heat loss, gas exchange and diffusion rates in animals. So in a large object like an elephant, it has a smaller surface area to volume ratio, so the rate of heat loss is slow, rate of diffusion is slow. Small objects such as this mouse has a large surface area to volume, rate of heat loss is fast, rate of diffusion is fast. Okay, so I'm going to post some resources for in-depth studies, so I'm going to put the links in the boxes below for the required practical videos, also links to exam packs, and they'll be below this video. Remember to subscribe, as week 2 I'm going to go through Organisation 1, which is digestion and enzymes. I'll see you soon.